Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on the gallbladder. Now, before I start this presentation lecture on the gallbladder, I would like to show you some figures, and these figures are very important for you. Now, as you can see on the screen, the lever is shown. In fact, the surface of the lever is the, as you can read in the bold, it is written diaphragmatic surface of the liver. Then the ligaments, then the gallbladder, left lobe, right lobe, inferior vena cava. Okay, now this is the visceral surface of the liver, and you see see the position of the gallbladder and the ligaments. Now here they are showing the black color arrow, and actually you see this part, this rectangle box, very small rectangle box. So we are zooming this rectangle box, and what this rectangle box is. So what is this? So all this hexagonal structure are the liver lobules, and around the liver lobules, as you can see, the portal triads are present and the branches of portal triad. Then in the center, central vein is present, and this is the information of this entire figure. So now this figure, blood supply to the liver, how the blood is supplied to the liver and the portal triad. So this is also a very good figure. Read all the names, see the position of central vein, then the portal triad, hepatic sinusoid, lymphatic vessel, and this is the information of the figure. And you know, in our previous lecture, the first point that we discussed, that the classic liver lobule. So here this is the diagram, this one is the diagram of classic liver lobule. You see the portal triad, then in the center there is a central vein, and this reddish brown color, these are the hepatocytes. Then you see green color bile duct is shown, then the hepatic sinusoid. Okay, and this is the information regarding this figure. Now some histological slide. This one is the photo micrograph of pig and human liver. Then comparison of three models of liver organization and a function. So in our previous lecture we had discussed about the classic lobule. Then second point in the previous lecture, portal lobule and the oval mass, hepatic acinus or the liver acinus. So you see the arrangement of these three components: classic lobule, portal lobule, hepatic acinus or the liver acinus. Then you also observe the central vein and where the portal triad are located. And on right hand side, two neighboring liver hexagonal shaped lobules are shown, and in between the this one, between two central vein, this is the hepatic acinus, and with the different color they have shown us the zones of the liver. So number 1 zone 1, number 2 zone 2, number 3 zone 3 of the hepatocyte. Long axis, short axis, then the triad, where the triads are situated. Then this figure, this is also very informative figure. See, here portal triad is shown, and with the white arrow the direction of the blood flow is shown in this figure, and with the green color they are showing the direction of flow of the bile. So as we can see in the figure that the right arrow and the green arrow, they are opposite. That means the flow of blood and the bile is opposite in direction. They are flowing blood and bile opposite in direction. Coffer cell. Okay. Then in the center, central vein is shown. Okay. Then this is the information regarding this diagram. Then this, in this figure, hepatic sinusoids are shown. This is the electron micrograph of two hepatic sinusoids of the liver. This one is the first hepatic sinusoid, okay, and this one is the second hepatic sinusoid. Then Coffer cell, hepatic microvilli, okay. Then this one, as you can see, this is the hepatocyte cell, this one is the hepatocyte, and we know the entire row of the hepatocyte is known as the lamina. So here on left hand side we can say this is the one lamina, and here this one is the other lamina. Between this two lamina there is a space, and this space is surrounded by the endothelial cell. So these are the endothelial or the epithelial cell, and this in between space of the hepatic lamina is this space is the lumen of hepatic sinusoid. And as you can see in the sinusoid, the blood, the RBC, we can observe the RBCs. So that means the blood is present in the hepatic sinusoid. So you go through this entire figure and the information regarding this figure. Okay. Now here electron micrograph of a hepatocyte, okay, in this figure. Now we are starting the presentation lecture on gallbladder. This is the first point of our today's discussion. Now here you can read this, you can go through this point, you can read this first point, and I am directly explaining this point to you. Now we know the cells of the liver, 80% of the liver area is covered by hepatocyte. This hepatocyte secrete bile. From the hepatocyte, bile enters into the canaliculi. Canaliculi opens into bile ductules, and bile ductules open into the bile ducts. Now, here I am drawing two lobes of the liver. This is the right lobe, and this one is the left lobe. This one is the right, this one is the left lobe. Now, right now I am drawing the bile duct. So here now you can see the bile ducts are opening into right hepatic duct, and here the bile ducts, they are pouring the bile into the left hepatic duct. Now this right and left hepatic duct, they merge together and they form common hepatic duct. So this is the common hepatic duct. Okay, this is the common hepatic duct. Now we know the bile is stored in the gallbladder. Now from the gallbladder, cystic duct is arising, and this cystic duct is merging. So cystic duct is merging with common hepatic duct, and one new duct is formed. So this new duct is known as the common bile duct. Now this common bile duct joins with the main pancreatic duct. Okay, and these two ducts, when they combine, they form short bulge like part, and this swollen short part is known as hepatic pancreatic ampulla, and this ampulla opens into duodenum at major duodenal papilla. So this is how the bile is poured into the small intestine, that is the duodenum. So this is the route of bile, how the hepatocyte are producing the bile, how the bile travel, and how the bile enters into the duodenum. So this is the first point of our discussion. Now moving on the second point. Now before that, I am zooming this figure, so you go through this figure. This was the theory point, and this is the entire figure for the first point. Moving on the second point of our discussion. Now, second point, first you observe this figure. The green color portion, the green color structure is the gallbladder. You observe the position of the gallbladder, where it is placed. So this is the second point. Gallbladder is hollow, pear shape. We know pear is a fruit, green color fruit. So the shape of gallbladder is the pear shape organ. The gallbladder is, and gallbladder is attached to the lower surface of the liver, and the gallbladder is capable of storing 30 to 50 ml of bile. And the important point to note over here, Not only the bile is stored in the gallbladder, but it is also concentrated during the storage. And you need to understand how the bile is concentrated inside the gallbladder. So, towards the end of this presentation lecture, I will explain you the entire mechanism how the bile is concentrated in the gallbladder. Now, you go through this figure. Then this figure. Now moving on the third point of our discussion. Now in the third point we are discussing wall of the gallbladder. Okay, histological slide, stain with hematoxylin and the eosin, and we are observing the wall of gallbladder under low magnification of the microscope. So theory point, third number. The wall of gallbladder consists of mucosa. This is the mucosa. Okay, mucosa. And mucosa composed of simple columnar epithelial cell. So here I am zooming. So you see the position of. So in the mucosa, simple mucosa is made up of simple columnar epithelial cell. Now just inferior to this simple columnar epithelial cell, lamina propria is present. So here this is connective tissue, areolar connective tissue, lamina propria, just beneath the columnar epithelial cell. Then muscularis with the bundle, thin muscularis with the bundle of muscle fiber oriented, directed in several directions. So these are all, as you can see, smooth muscle fiber, and they are scattered, they are present in all directions. So smooth muscle fiber. Then the outermost layer, the external layer, is the adventitia or serosa. So in this figure the serosa is shown. Okay, the outermost layer, serosa. So now I am showing you the entire slide. You go through all the names, try to remember as many names as possible. Now here one more, see, I have repeated the third point, and you see this is another histological slide. First layer, the entire mucosa layer. Okay, this is the mucosa composed of simple columnar epithelial cell and followed by lamina propria, and here muscularis externa. So if you observe closely, Here you can find the smooth muscle fibers. Here these are all smooth muscle fiber. Okay. Muscular is extreme. Then the outermost layer in the previous slide, serosa, they had shown to us, and here the outermost layer, adventitia. So I am zoom out so that you can see the entire figure properly, and this is the information regarding the figure. Now the fourth point, mucosa has numerous folds. Mucosa is having many folds, and mucosa folds are particularly evident. That means the mucosa folds are clearly seen when the gallbladder is empty. So you follow this black color arrows, this arrow and this arrow. So these are the mucosal fold. So for example, this one is the mucosal fold, this one is the mucosal, these all are the mucosal fold, and they are clearly seen when the gallbladder is empty. Now moving on the fifth point. Now we know the mucosa is composed of simple columnar epithelial cell. So here in this slide they are showing, this is the electron micrograph of simple columnar epithelium of mucosa, and first you observe this figure, then I am explaining the entire thing. Now here see the lining epithelial cell. Now what is the meaning of lining epithelial? So lining means, here imagine this is the lumen, this one is the lumen of the gallbladder, and in the lumen the bile is present. Okay, so the cells surrounding the lumen, they are the simple columnar epithelial cell, and this cell are known as the lining epithelial. Now what is there in this cell? So prominent mitochondria, many mitochondria are present in this cell. Then on the periphery, the plasma membrane or the cell membrane is having many finger-like projection, and this finger-like projection are known as the microvilli. So as you can see on the surface of the cell, microvilli are present. Then large intercellular spaces between the cell. So here this is the one cell, this one is the one cell, this one is the one cell, second one, this one, this and this. Then here between the cell with the arrow, this space, and over here this space. This is the intercellular space. Okay. Now what happens in this intercellular space? So this intercellular space are going to fill with water, and how water is reaching to this intercellular spaces. So from bile minus water, from bile water is removed. So that means bile is concentrated. And what is the mechanism for the concentration of the bile and where the water is going? That is the question. Okay. So here, now the mechanism for the concentration of bile involves activity of the Na plus pump, where this Na plus pump are present. So in the basolateral membranes of the epithelial cell, columnar epithelial cell. Then active transport of Na plus, and then this active transport of Na plus will be followed by passive movement of water. So the bile is concentrated. So entire mechanism I am explaining at the end of this lecture. So this is the sixth point, fifth and sixth, both the point. Moving on the seventh point. Now this is the seventh point. In the seventh point, here the green color pear shaped gallbladder. Inside the gallbladder, bile is stored. So this is the bile they are showing. Okay, this is the bile which is stored in the gallbladder. Now what is happening? How this bile is going to enter into the cystic duct? How it will enter into this cystic duct? So see, duodenum. In the duodenum, the wall of duodenum, that the proximal duodenum, enteroendocrine cells are present, and they secrete this hormone cholecystokinin. So this hormone is poured in the blood. Via the medium of blood, the CCK reaches to the gallbladder, and this CCK is affecting the wall of the gallbladder. So the wall of gallbladder is contracted and the stored bile is pressurized. So one factor is the stored bile is pressurized due to the contraction of the wall of the gallbladder. And the second thing, here one sphincter is there. We know this one is the body, okay, and this is the neck region. So here the region from where the cystic duct is arising, from where the cystic duct is originating, here one sphincter of smooth muscle is present, and this CCK is relaxing this sphincter. So the bile enters, so the bile can enter into the cystic duct. Okay. So this is how the CCK is contracting the gallbladder. CCK is relaxing the sphincter, and the stored bile enters into the cystic duct. From cystic duct, it will enter into the common bile duct, and this common bile duct merge with the pancreatic duct. Then ampulla, hepatopancreatic ampulla, and ampulla is going to open into the duodenum, and this is how the bile is poured into the duodenum. So this is the seventh point. I am showing the entire figure. This is the theory point, and you observe the figure. Now moving on the eighth point. Now this is the eighth point. Now question is how the release of CCK is stimulated, which is the factor why CCK is produced by the enteroendocrine cell of the duodenum. The reason is, see, imagine you had eaten the pure ghee paratha in your lunch, and the pure ghee paratha is having a fat in the form of ghee. When this ghee or the fat reaches into the duodenum, it stimulates the cells of the duodenum, that is the enteroendocrine cell, and this enteroendocrine cell, they release CCK. Now what this CCK is doing, so now here CCK is performing the third function. Already we have discussed the two function, and here we know this is the hepatopancreatic sphincter, and this sphincter is composed of smooth muscle. So this CCK is relaxing this hepatopancreatic sphincter. So the smooth muscle of the sphincter, they dilate, they relax. So hepatopancreatic ampulla is opening into the duodenum, and the bile is poured into the duodenum. So all this point, all this information, all these three action of CCK are written over here in the 10th point. You can go through this point. I have explained this point. Okay, the three action of CCK. Then now this is also very good information for you. Only one page is there, and this one page covers the entire topic of the gallbladder. So you can go through this information. Then point wise they have written 1, 2, 3. Okay. Now one mechanism. Now one question is there that I need to explain you. How the bile is concentrated. How the bile is concentrated, this is the question. Now what is concentration? Bile minus H2O is equal to concentrated bile. That means removal of water from bile, then this bile is going to be concentrated. What is the mechanism? This is the these are the simple columnar epithelial cell. For example, very large cell, this one, because they are columnar one, this is one cell, and you see how long it is. This is here, the cell is ending. This is one cell. This is the neighboring cell, second one. Okay, it is also very large. See, this is the second cell, and in between this white color portion, this is the intercellular space. This is the intercellular space. Now what happens? Imagine here, this is the bile, okay, and in the bile there is water. Now this membrane, this part, this is, right now I am showing you the basolateral membrane. This is also the basolateral membrane of the neighboring cell. Now in this membrane, this is the cell membrane. So in this membrane, this is the ion channel, okay, or the pump. This is the Na plus pump or the ion channel, and ATP is providing energy to this Na plus pump. So this is the cytoplasm. In the cytoplasm, Na plus is there. So active transport mechanism, and with the active transport, this Na plus are pumped out. Na plus are positively charged. So due to the attraction, Cl minus are also following the Na plus. So in the intercellular space, what is happening? NaCl is there, and here is the bile. So these water molecules, they are attracted towards this NaCl, and the water molecule, they are moving passively. Now from the intercellular spaces, H2O, where this H2O is going? So H2O will enter into the connective tissue. From the connective tissue, where this H2O is going, so now this H2O from the connective tissue, it will enter into the blood vessel. So this is how the bile is concentrated. Now one more figure is there. Now before that, I would like to show you this entire figure. You go through this figure this way, and the entire mechanism that I had explained you is written in this figure. Okay, here the mechanism is written, so you can go through this, and this is the final figure. This is also very good one. See, first point, duodenal cell secrete hormone cholecystokinin and secretin. This two hormone enters into the blood. So here the duodenal cell are shown, and from the cell you can see the secretin is poured in the blood, CCK is poured in the blood, Now this CCK, you see this CCK, you follow this black arrow where this is going. So this CCK is acting on gallbladder. What CCK is doing? CCK triggers contraction of the gallbladder, so the gallbladder releases the bile. This is one thing. Okay, then what this secretin is doing? See, from the gallbladder, bile was released, and in the bile there is a bile salt. So now in the blood vessel there is two things: bile salt is there and the secretin is there. So these two factors combinedly, the bile salt as well as the secretin, they are producing the positive feedback mechanism on the gallbladder. So due to this positive feedback of secretin and bile salt, gallbladder releases more and more bile. Okay, so this is the last figure of today's presentation lecture. I am zooming this so that you can see this figure properly. Okay, and with this figure we have completed the discussion of the gallbladder. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.